everyone. Thank you for the opportunity to speak here today. And what I want to discuss today is how proteins are distributed and transported within reticulated, networked organelle structures. So broadly speaking, my group is interested in exploring intracellular transport. So as a cell, how do you robustly and efficiently deliver everything from ions to proteins to organelles to where they need to go within this very complex cellular environment? And this includes looking at regulation of motor-driven active transport, as well as diffusive transport in these geometrically complex and physically complex cellular structures. And we do this with a combination of computational modeling, coarse-grained simulations, and live cell imaging data that we get from collaborating groups. So today I want to talk about two different sort of stories on this topic. In particular, the relationship between morphology and protein distribution in the endoplasmic reticulum, and the diffusive transport of proteins and other particles within reticulated organelles. So jumping into the first one, this is work that we're doing in collaboration with Laura Westrade's group in Calvin University, looking at how the structure of the ER affects protein distributions. So for a bit of cellular background here, the endoplasmic reticulum is this morphologically complex structure. It has stacks of sheets near the perinuclear region of the cell, and it has an extensive network of interconnected tubules out in the cell periphery. And they all share one continuous lumen. They have multiple functions, biological functions for this organelle, everything from calcium storage to lipid distribution to the folding and quality control and export of proteins down the secretory pathway. Now, of course, when we look at one of these beautiful images of ER structure, what we're actually looking at is the distribution of some fluorescently labeled ER protein. It's a membrane protein in this case. And so one of the first things we wanted to ask was, how can we quantify that distribution? How can we get out some kind of information about morphology and where the proteins are going from looking at those images? And the first time we tried to do that, it didn't work, in the sense of when we took the known accepted measurements for ER tubule radius from EM measurements, about 20 nanometer radius for the tubules, we found that the tubules we saw were far too bright. They're far too bright for something that is that narrow, which quickly led to the realization that, in fact, we have enough information here to estimate the local morphology of the ER. So if we look at the relative brightness of tubules, and I'm sorry, this projector doesn't project it very well, but there are tubules underneath those blue guys there, and a nearby peripheral sheet region, the relative fluorescences of those for a membrane protein should tell us how wide the tubules are. And because those relative fluorescences are going to scale with the area of the sheet, the length of the tubule, which we can measure, and the radius of the tubule. So from there, we get an estimate of tubule radiuses of about 53 nanometers. It's a good deal bigger than was previously estimated from EM data. But very recently, there's been super resolution microscopy studies that came out that got very similar radius measurements, right around 50 nanometers. And so using just confocal microscopy, very easy to obtain images where 50 nanometers is well below their diffraction-limited resolution, we can estimate the radius of the tubules fairly accurately. And then we can do the same thing using a luminal protein to try to estimate the height of the ER sheets, the separation between the two membrane sides of the sheets. We get heights of about 40 nanometers or so, again, consistent with what's seen in super resolution images, although even there, they don't have the resolution to quite tell how far apart the sheets are. All right, so once we have that technique in hand, if we can look at the relative fluorescences of luminal and membrane proteins in the tubule and in the sheet, we can then start to see how those change if you perturb the cells. So the Westrade group has the ability to overexpress certain proteins in the ER. So in particular, they overexpressed RTN4, which is a protein that stabilizes ER tubules. And it's been known for a while, if you overexpress that, then the tubules proliferate. You get more tubules in the periphery. But what we found also is that the individual tubules become narrower. They narrow down by about 30% when RTN4 is expressed. Similarly, we can overexpress a protein called Klimt63. It's known to stabilize ER sheets. It wasn't originally known whether those proteins pull the sheets together or act as posts, pushing them apart. But it is accepted that when you overexpress Klimt63, the sheets proliferate. You get more sheets in the peripheral region. So we found using our quantification approach that Klimt63 actually increases sheet spacing. So if you overexpress it, your sheets become about 40% further apart, indicating these are acting as posts to keep the ER sheets apart from each other. All right, so now we can actually flip that argument on its head. And instead of going from relative protein fluorescences to the morphology of the ER, we wanted to point out that you can go the other way around, right? This means that if you change the ER morphology, you're going to change the relative concentrations of membrane and luminal proteins. You're changing the surface area to volume ratio, effectively. So when you overexpress RTN4, you are decreasing the radius of the tubules. And the radius of the tubules is going to be proportional to your luminal versus membrane proteins. So you're going to be changing the ratio of those by about 30% when you overexpress RTN4. If you overexpress this sheet stabilizing protein, you're going to change that ratio of luminal to membrane proteins by about 40%. And so there's this intimate connection here between the local morphology of the ER, the surface area to volume ratio, and the stoichiometry of the proteins, which means if you have any kind of reactions that combine membrane-bound and luminal proteins, the relative concentrations of the reactants are going to change when you change these morphologies. All right, so this is work that's still very much ongoing. I just wanted to bring it up here as an example of information you can get from quantifying these fluorescent distributions. But now I want to switch over to talking about some more dynamic questions. So how do proteins or other particles get around these network structures? And here we're going to think about both ER network structures that we get from Laura's group, Laura Westrait's, and mitochondrial networks, which we have obtained in collaboration with the Rafalski group at the Allen Institute. And all of the computational work I'm going to talk about was done by a postdoc in my group, Hayden Brown. Right, so the big question here is, how does the structure of the network affect the efficiency of diffusive transport of proteins and ions through these organelles? Now, to give you a bit of background on diffusive transport in complex media, this is usually thought about in the context of a continuum fractal type of medium. And a fractal has one characteristic dimension. It relates how many sites are available to you, n, versus the spatial extent of your domain, r. And that fractal dimension will affect qualitatively how diffusive search behaves. So if you have a dimension greater than 2, you're in a regime of bulk kinetics, or non-compact search. So in that regime, your search time for any part of your network scales linearly with the number of sites available to you. You have a sparse exploration of your domain. So there's a little particle diffusing in 3D here, and it's leaving lots of holes behind. And you have a tendency to explore the whole domain, to hit the boundary before you find any target, regardless of where you start in that domain. By contrast, if you have a domain of dimension less than 2, you will be in the regime of compact search, where you have geometry-controlled kinetics. So the closer you start to your target, the more likely you are to find that target before you ever hit the boundary of the domain. And your search time is going to be some stronger dependence on the number of sites available to you. All right, now, of course, this sort of fractal continuum explanation can be somewhat limited in explaining actual cellular structures because they're not fractal. You can't zoom in on them forever and get the same sort of structure. There's a cutoff length, and there's, in terms of the edge length for the smallest cutoff, and then there's a cutoff length for the size of the cell itself. And so we wanted to look more explicitly at an actual network structure. So how is the structure of that network going to affect diffusive search? And we did this using a fairly standard metric called the mean first passage time. So this is the time to get from any one point in your network to any other point if you are a diffusive particle, as an average overall possible paths you can take. So we have an analytical approach to calculate this mean first passage time. Basically, we use the results from one-dimensional diffusion to find a propagator matrix, the distribution of times to get from one node to each of its nearest neighbors. And then we can convolve over many of those propagators, sum over all possible paths you can take, and find exactly the average diffusive time to get from one node to another. It turns into a rather simple matrix inversion problem. All right, so with that tool in hand, we can do things like take an ER network structure. So this one right here was extracted from a fluorescent image. And then say if we have a particle that starts at a certain node of that network, how long on average until it gets to all of the other nodes. And as you can see, this is a characteristic of compact search. The further you are from the starting point, the longer it will take you to find those nodes. And even for some nodes close to your starting point, you have rather high search times because they happen to be poorly connected to the rest of the network. So there is some heterogeneity as well in terms of how easily you can find the different nodes of the network. All right, now because we have an analytical approach of getting these mean first passage times, we can ask some fun questions about optimizing over network structures as well. So the simplest sort of optimization problem we could come up with is an issue of peripheral delivery. So suppose you have a bunch of particles starting in the center of the cell, and you have a bunch of targets scattered throughout the cell periphery. And you want to design a network to diffusively deliver particles to each of those targets as quickly as possible. So we want to minimize the mean first passage time of particles to each of those targets in the periphery. Now you might think the simplest thing we could do would be to connect the bulk of the cell, the center, to the peripheral targets. And we can put in some estimated numbers for the size of, say, a yeast cell, the typical diffusion coefficient of a protein in the ER. You'd find that mean first passage time on the order of a minute or so. However, this sort of network is actually quite inefficient. In particular, if you're a target out there waiting for a particle to arrive, and that particle happens to go down the wrong path, it'll have to go all the way through the cell to find you again. So you can speed things up quite a bit by connecting the peripheral targets themselves, so building more of a network out on the periphery. And then you can speed things up some more by getting rid of all of those excess connections to the bulk of the cell, leaving one connection from the bulk out to the periphery, and then having a well-connected lattice on the periphery of the cell. This turns out to be the fastest network structure we can find. And conceptually, it makes a lot of sense, right? You want to get particles out of that bulk region to the periphery, and then they can explore a lower dimensional space out on the periphery. Interestingly, if you look at the structure of the endoplasmic reticulum in yeast cells, it is very similar to that sort of optimal structure. You have endoplasmic reticulum near the nucleus, you have a few, one or two connections out to the periphery, and then you have a well-connected lattice out in the periphery of the cell. All right, now we can take this one step further and consider now what if we have multiple targets that particles are trying to find. So a particular biological application might be proteins searching for exit sites in the endoplasmic reticulum. So for a protein that's translated into the ER to leave the endoplasmic reticulum, it has to find one of a number of punctate scattered exit sites. And so we ask the question of what will happen if we take our ER structure, we scatter many target sites, and then we look at the overall rate, how long until you've hit the first of those exit sites as a function of the number of exit sites available. So that's plotted here. Here's the exit site density and the search rate. And you can see when the exit site density is very low, you get a search rate that's proportional to the density, just like you would expect for bulk kinetics, rate proportional to concentration. But when the density gets much higher up here, you start seeing what you would expect for a one-dimensional search. Your search rate becomes proportional to the concentration squared. And if we look at the physiologically relevant distributions of exit sites, oh no, then it doesn't show up on your screen. That's amazing. All right, they're around between 1 and 6 or so per micron. So it's in the region where you're already starting to deviate from the linear scaling, deviate from bulk kinetics. Incidentally, in terms of actual time scales, we find on the order of about a minute for a protein to just diffusively find the first of an exit site out, the first exit site in those scales of density. Okay, so to take this one step further still, we wanted one single metric for how easy is it to explore a network by a diffusive process. And the easiest single metric we could pick is a target averaged global mean first passage time. So you look at starting on one node of the network to get to any other node of the network, what's the average time for that to happen, averaged over all starting positions, averaged over all target positions. So you get one number for a network. And there's an example here of two networks covering roughly the same spatial extent. This one has a very low target average global mean first passage time. There's not very much edges to explore, and they're all very well connected. This one has a very high search time. There's a lot more of it to explore. The edges are poorly connected. So we can take that metric now and apply it to biological structures. In particular, we got hundreds of structures of mitochondrial networks that were extracted from many cells by Susan Rafalski's group. She's currently at the Allen Institute. And so they have these mitochondria. They tend to be on the periphery of a cell. They can map them to network structures. They also had mutant cells where they mutated the fission and fusion proteins responsible for mitochondrial dynamics. And those mitochondria still form networks, but the connectivity is different. So these networks tend to be much less well connected. And so we wanted to ask, how does these different network structures affect the diffusive transport of particles? All right, so plotted here is our search time metric versus the total edge length in one of these mitochondrial networks. Obviously, the more extensive the network, the longer it would take to search. Each little dot here corresponds to a single wild-type network for a blue dot and a single mutant network for a red dot. And the main feature to see here is that the mutant networks do, in fact, tend to have longer search times than the wild-type networks, even for the same total length of network. So there's something about that reduced connectivity that is, in fact, going to make it harder to search through these networks. All right, so we can quantify that using a metric from graph theory called the cyclomatic, or the loop number. It's easy enough to calculate for an arbitrary network. It's the number of edges minus number of nodes plus 1, and it computes the number of independent loops in a connected graph. So plotted here are all the same mitochondrial networks with the total edge length, the loop number, and then the color corresponding to the search times. So as you would expect, again, as you increase the total edge length, the search time increases. As you increase the loop number, the search time decreases. And interestingly, for the same total edge length and loop number, the wild type and mutant networks have very similar colors, very similar search times. So those two parameters seem to be mostly responsible for the difference between those two network types. And we can make that a little bit more formal by looking at synthetic networks. So if we now generate lots and lots of honeycomb network structures at different densities, different numbers of edges removed, we can thoroughly explore the whole space of total edge length and loop number. And we'd find these nice, smooth dependencies on these two parameters of how long it would take to search through the network. And in fact, I won't get into the math here, but you can map this to a percolation problem and show that this dependency is pretty well described by a rather simple scaling law here, involving the total edge length and the loop number. Right, interestingly, each of the bins here has hundreds of networks in it, so we can find a coefficient of variation. So if I specify edge length and loop number of a network, I can then ask how much variability is left in the search time on that network. And we get coefficients of variation up to about 25% or so. So within plus or minus 25%, you can predict how hard it is to diffusively search through a network, given these two simple morphological parameters. And we can then go back and apply these to the biological networks. So here, for all the different mitochondrial networks, for a given number of loops, for a given edge length, we can then compare the search time on that actual network structure compared to what we would expect from that simple scaling relationship I showed earlier. So the ratio is plotted here. There are, of course, a few outliers, but for the most part, it's centered around 1, plus or minus 25% or so. Similarly, you can do that with pieces of an ER network structure. They look very different, much higher loop numbers. But on the whole, you can predict the search time just based on these two simple morphological parameters. All right, and finally, to get at a metric that's of perhaps more direct biological relevance, we also wanted to look at kinetics. So now not just searching for a particular target, but what if you have multiple diffusing proteins or particles in there? How quickly will they find each other? So for this, we resorted to simulations, and we have some very efficient methods for simulating diffusion on a network structure where you can propagate the particle quickly from node to node. And what we do is we simulate pairs of particles. We get a distribution of search times for those pairs, and from there we can extract an overall search rate as a function of particle concentration. So that's plotted here, the ratio of the search rates on wild-type versus mutant mitochondrial networks. And you can see that if you have particles at high concentration, the network structure doesn't matter. You're going to have a reaction rate that's quite fast, just based on how quickly particles find near neighbors in the same tubule. If you have particles at low copy number within these networks, low concentrations, below about 1 per micron or so, then that overall network structure is going to have a big role to play. In fact, you can get up to about 80% speed up in these wild-type networks compared to the mutants. So overall, then, if we're talking about proteins with fairly low copy numbers, and there are proteins in mitochondria known to have copy numbers that low, then your mutant networks are expected to have substantially slower kinetics just based on the lower connectivity of that network. All right, so to sum it up then, we've explored the connection between the local organelle morphology and the ratio of membrane and luminal proteins in the ER. We've shown that the diffusive search time on networks can be fairly well predicted by a combination of these two morphological parameters, the tube length and the loop number. And we've shown that mitochondria, with their fission and fusion mutants, can actually alter kinetics by changing network connectivity. So I'll stop by thanking the people involved in this work. So all the experimental data came from these two groups here, the Westrade group and the Rafelsky group. And Aiden's a talented postdoc in my group who did a lot of the computational work. And thank you for listening.
Questions?
The mean first passage times seem rather small. Are these actually really significant for the evolutionary fitness of the cell?
Right. It depends on which biological process you're thinking about, right? Because kinetics are going to be proportional to those mean first passage times. So that means any reaction rate in the ER is going to be proportional to those mean first passage times. And they are in the tens of seconds to a minute regime for the mammalian ERs. So I would say, that is comparable to a lot of the kinetics seen. It takes maybe a few minutes for a protein to leave the ER altogether.
Yeah. So you pointed out that the rather sparse networks actually have low mean first passage times. But another possibility is that the cell is just minimizing the investment of material put into the network.
Yeah, so that is an interesting point. There's been a lot of research on transport by pumping with potential differences in networks where they worry about what's the best you can do with a limited amount of network material. And we could put that particular limitation in these models. For now, we didn't, because I think it's one of the interesting features of diffusive networks is that there are optima even when you're allowed to use all of the networks you want. You can put in more edges, it won't help you. In principle, you could put in arbitrary limitations in terms of some kind of a linear penalty or a quadratic penalty and how much network length you make. But we don't know how the cell feels about spending more material to make networks.
Yeah, thanks.
I have a question, which is what about transport mediated by networks breaking apart, moving somewhere else and refusing somewhere else? Like, can you get sort of leapfrog over the rest of the network that way by fission and fusion?
Right. So when we started out, we really wanted to go down that road, the dynamics of the ER rejoining to different positions. As far as we can tell for now, though, it's very slow compared to these diffusions. So within a minute, maybe you'll have a few tubules shift and not very far. And so for these particular timescales we're looking at, I don't think it's going to be very significant. Yeah.
Maybe I missed it, but is there some limit in which you can understand analytically why it's only loop and length that matter? I mean, looking at these things, you might think, I don't know, that I move this edge over there and somehow it should make a difference, but.
So it is sort of a result on the average. Obviously, you can construct really weird networks where this is the loop number and the edge length are the same, but they have a very slow diffusion time. Like disconnect into two pieces and leave one here. Yeah. But as long as things are fairly spatially homogeneous, it doesn't seem to matter very much. That's what we were trying to get at with all of those honeycomb networks. Right? We generate them randomly, and they all fall within sort of 25% of the average there.
So is there a calculation in which that 25% would sort of approach zero? I don't know if the coordination number became larger. I don't know, just some way of understanding why those details fall away.
Right, so I think the best why we have so far is the argument about the percolation scaling. You can treat it as a percolation system. It has that universal scaling exponent to it. And I think it's, vaguely speaking, it applies whenever your nodes are sort of uniformly distributed through space. It's a homogeneous system, but we haven't been able to quite formalize the limit on that part of it, yeah.
I have a question which might be considered somewhat nitpicking, which is the nature of the average. In some contexts, it might make more sense to average the rate rather than to compute a rate by taking 1 over the average of the first passage time.
Yes.
Is this just a trivial matter, or is there any substantial difference?
No, it's not a trivial matter, right? Because all it would take is one particle that is incredibly slow to make your mean first passage time huge, but maybe not affect your average rate very much. So that's one of the reasons we started looking at these sort of more kinetic metrics, right? Because here we take the full distribution of search times, and there you actually are getting, like, by looking at that whole distribution, not just the average, you are getting actual kinetic rates directly from that. But for these, we rely on simulation, so there's a limit to how much calculation we can do with that.
So when you estimate the dimensions of the ER tubules and sheaths, do you assume the concentration of your labeled protein is the same everywhere?
Yes, yeah, that's a great question. When we say the radius must be this, that assumption is underlying it. Originally, we wanted to start with the morphology and say, can we show whether the concentration of proteins is homogeneous or not? We quickly realized that wasn't going to work from what people thought of the previous morphology. So now we're going backwards. We're saying, suppose they're homogeneous. This is what the radius would have to be. Now we've measured it with several different membrane proteins and different luminal proteins, and we get the same radii and sheet separations, which is indicative that it's probably not one particular protein concentrating strangely in one versus the other.
Okay, so thanks for a great talk.